Here are the learning goals that we will accomplish as we proceed through this lecture. So we're going to start with talking about the differences and similarities between skeletal, cardiac, and smooth muscle. So I gave you guys this table. It's a great table that compares skeletal, cardiac, and smooth muscle in terms of its body location, the cell shape, and the appearance. So skeletal muscle, as many of us know by now, thanks to the tissues section, is the muscle that is attached to bones, or some facial muscles are attached to the skin. So it's muscles that are attached to either bones or skin. In terms of its histology, what it looks like are these long cylindrical multinucleate cells, which means there are many nuclei, as you can see by all these little dots shaded in this darker purple color. And we've got obvious striations here. All right, and so this is an actual histology slide, and then sort of a 3D animated what it would look like based on this slide. Because we know histology is 2D, so it just makes it easier if we can see it in a 3-dimension. The second type of muscle is cardiac muscle, and this is located within the walls of the heart. So the cardiac muscle is what it's contracting, and because this actual muscle is contracting or getting shorter, it forces blood inside of the heart to pass through its multiple different chambers. And in terms of histology, what we see here is some branching chains of cells. They're either uninucleate or binucleate, and we can see obvious striations occurring as well in this plane. And so you can see here would be the end of the cell. As it moves down, we can see branching occurring. Excuse me. And so finally, the third type is the smooth muscle, and smooth muscle is the muscle that is located in the walls of our hollow organs other than the heart. And there's also multi-unit muscle that is in our intrinsic eye muscles, our airways, our very large arteries. And in terms of its histology, it looks like these single sort of spindle-shaped, and as you can see, here's our spindle shape, uninucleate, and there are no striations that are actually within smooth muscle. So you might find smooth muscle, like I said, in your arteries. You would find it along the GI tract, in the bladder, to some extent in the alimentary canal as well, in the airways. Okay, so these two, cardiac and smooth muscle, are involuntary muscle, whereas skeletal muscle is voluntary. We decide when we want to, for example, contract our biceps muscle. So it's under conscious control, whereas cardiac and smooth muscle are involuntary muscle, meaning that we don't have control over their contractions. We don't tell our heart that we want it to beat faster and it just does it, okay? So it's involuntary, sort of an unconscious contraction. And same with smooth muscle. So again, as we move through this lecture, here is skeletal muscle. I won't really necessarily go through everything that I just went through. The only other thing that I will point out here with skeletal muscle is that it is very fast contracting, but it does tire easily. Alternatively, it is a very powerful contraction. So it's rapid, so it's a fast contraction and a powerful contraction. However, it does tire easily, which is why you can't just do 1,000 push-ups at free will unless you're really fit. Because if you can do 1,000 push-ups at free will, more power to you. And then here is just basically everything that I already said about the cardiac muscle. It's found only in the heart, makes up the bulk of the heart walls. It is striated and involuntary. Finally is the smooth muscle, and I'm just going to briefly talk slightly more about this. It is in the walls of our hollow organs like the stomach, the bladder, the airways, but it's also found in the walls of our arteries and our veins. So we know that sometimes our arteries get bigger and sometimes they get smaller. For example, when you're cold, you think of your fingertips and your toes, for example, tend to get cold faster than the core of your body. That's because the vessels in your fingertips and your toes actually get smaller. They vasoconstrict in order to preserve heat and redirect heat to the center of your body. And so the reason that our vessels are actually able to get smaller is because they have muscle in them. These are very elongated cells, as you can see. They're non-striated, so they don't have the up and down stripes like we saw in cardiac and skeletal muscle. There's only one nucleus in each cell. Again, involuntary, meaning we don't have conscious control over when this type of muscle contracts. And these contractions tend to be very slow and sustained contractions. All right, so think about moving food through your GI tract, for example. You don't eat, it passes right into your GI tract and then comes out. It's a very slow process, they're very slow, sustained contractions that are propelling the food through the those hollow organs in the GI tract. The idea that there's a lot of blood that is supplying smooth muscle. And most smooth muscle is going to be organized into sheets in the walls of all the vessels, except for capillaries. And then it's also organized into sheets in the walls of the hollow organs. And so there are two primary layers. When I'm talking about sheets, that usually I'm referring to the fact that there's really two layers occurring here. So we have one layer called the longitudinal layer, which is going to run parallel to the organ. So when the longitudinal layer contracts, the organ itself as a whole is going to become shorter. Okay, so longitudinal layer would run, the fibers would run in this direction. Okay, along the length. Alternatively, in the circular layer cells are actually running around the circumference of the organ, and so they are running literally like a clock around in a circle. Okay, so that's why it's called the circular layer. And so as you can imagine, when the cells in this circular layer contract, the middle hollow space called the lumen is going to get smaller. All right, it's like taking, I don't know, say a hose and squeezing it, and so now it's a smaller inside open area called the lumen in the human body. Interestingly, some smooth muscle has no nerve supply, and so it relies on hormones, on histamine, also on excess carbon dioxide, and lack of oxygen. So for example, gastrin is a hormone that stimulates the stomach muscle to contract so that it can churn food more efficiently. Another sort of unique characteristic about smooth muscle is that it can lengthen and shorten more than other types of muscle due to the tone of this muscle. Also the fact that contractions occur in very slow, prolonged periods. Additionally, this type of muscle doesn't require a lot of energy, and so this makes it capable of producing these extreme lengthening and shortening. So here would be a relaxed smooth muscle fiber, and then a contracted smooth muscle fiber. And you can see it's a pretty significant difference in its contracted and relaxed phase. Additionally, we have this stress relaxation response. And so, for example, basically this is describing the greatest example that I have for you is in the bladder. When the bladder is full, it allows the muscle to actually relax a little bit, which will allow some more stretch, thus being able to hold more volume. So these are just some cool, fun facts about smooth muscle. So in terms of muscle in general, all muscle tissues have 4 characteristics. So when I say all muscle tissue, I mean skeletal, cardiac, and smooth muscle. They all share these 4 characteristics that I'm about to talk about. So first and foremost is contractility. And contractility just means the ability of a muscle to shorten whenever it's stimulated. So here we have a relaxed muscle, and when we are in the contracted phase, so when the muscle gets stimulated, it shortens. Additionally, all muscle types are excitable or responsive, so they receive input from the nervous system, which allows them, basically gives them directions on what to do, right? So the nervous system in general is the main operator, and everything else just does what the nervous system tells it to do. And muscle is really no different. It responds to the nervous system to stimuli. Additionally, all muscle types are extensible, which means they can be stretched. Okay, so they can get longer if necessary. And then finally, all muscle types are elastic. And so that just means they're able to reform their original shape after they've been stretched. So it's kind of like a rubber band. When you stretch the rubber band, it goes back to its original shape and its original tightness for the most part. Muscle is really no different. It's elastic. So here's my first clear question for you guys. Go ahead and take a second and answer this one. And if you need more than a couple seconds, just pause this video so you don't get the answer automatically. Really try to quiz yourself. Alright, so now we're going to talk about 4 important functions of muscle tissue. This is pretty straightforward. Muscle tissue is responsible for producing movement. Muscles allow us to maintain posture and also our body position. Muscles help stabilize our joints, which we should know hopefully already based on the joints lectures that we've already covered. And then finally, muscle tissue, whenever it contracts, because it's burning energy, it creates heat as a byproduct. So muscle tissue is responsible and has the function of generating heat during contraction. An example of this would be, for example, if you're outside and it's pretty cold, you start to shiver. Okay? Shivering is basically the result of your muscles contracting. And the reason you're shivering is because when your muscles contract, they give off heat, therefore warming you up. So hopefully you learned something new already. Okay, so now is the point in the lecture where pretty much from here forward I'm going to focus mostly on skeletal muscle. So we're going to start talking about some microscopic and macroscopic features of skeletal muscle, and we will move on from there. In terms of skeletal muscle, we have three major features that we're going to talk about over the next 3 slides. So we're going to talk about the nerve and the blood supply. We're going to talk about connective tissue sheaths, and then finally we're going to talk about attachments. So nerve and blood supply. Every single muscle in our body is going to receive a nerve, an artery, and veins. Arteries and veins are present between what are called fascicles, and essentially a fascicle, so this whole pulled out section here, the entire thing is a fascicle, and there are multiple fibers inside of one fascicle, and there are multiple fascicles inside of a muscle. So in between these fascicles, which you can see these little blue dots in this picture, well, hopefully you can see them, is where we're going to find arteries and veins. Now, nerves, however, supply every single muscle fiber to control the activity of that muscle. So, remember, this is a whole muscle which is full of many fascicles. If we pulled out one of those fascicles, inside of it we would find many different muscle fibers. So we're going to pull out one of those muscle fibers. Every single one of these muscle fibers has a nerve supplying it. Contracting these muscle fibers requires an immense amount of oxygen and nutrients, and also waste products need to be removed quickly, which is why it's convenient to have the blood supply close, nearby, and present. In terms of connective tissue sheaths, every single skeletal muscle as a whole, and each muscle fiber and each fascicle is covered in a connective tissue sheath. And basically this just helps support the cells and reinforce each subsection and the whole muscle. And so in terms of the names of these different sheaths, the epimysium has the job of surrounding the entire muscle. If we pull out one of these fascicles, we will find the perimysium has the job of surrounding each fascicle. And then remember, inside of fascicles we have muscle fibers, many muscle fibers. And so if I pulled out one of these muscle fibers, this is what you see here, you're going to find the endomysium, okay? Sometimes called endomysium. I call it endomysium. To each your own. So we have the epi, the peri, and the endo, which are very common terms to us, hopefully, at this point. Anything with epi usually means outer, endo is usually inside, and peri tends to be sort of in the middle. Okay? All right, so now we need to talk about the attachments. So muscles, as we know, hopefully, we sort of deduce this from our joints lecture, but they span across different joints and attach to bones. And they attach to bones in at least 2 places, and it doesn't have to be the same bone. All right, so I'm going to give you the example here of the sternocleidomastoid muscle. All right, and so just to kind of give you a physiologic reference, anatomic reference of where we are in the body, if you turn your head to the side and you look at the front of your neck, you'll see a big, long, stringy looking thing sort of pop out of your neck right next to your clavicle, right next to your collarbone. All right, this is your sternocleidomastoid muscle. And in terms of skeletal muscle attachments, all skeletal muscles have a insertion, which is the attachment to the movable bone, and it has an origin, which is the attachment to the immovable or the less movable bone. All right, so let's talk about the sternocleidomastoid muscle. The origin of the sternocleidomastoid, it happens to be the sternum, which is where we get the sterno, the clavicle, which is where we get the cleido in sternocleidomastoid. All right, so these are the origins because these two bones, whenever we contract this muscle, these two bones aren't really moving. Their purpose is just to kind of stay put and anchor this side of the muscle down. The insertion happens to go onto the mastoid process, which is located on the temporal bone, if you guys remember from lab. And the insertion, again, refers to the bone that's going to move whenever the muscle contracts. So when the muscle contracts, it gets shorter. You can imagine it's pulling down this way. That turns our head that way. All right, so let me just kind of draw this in really quick. If this muscle shortens, it's going to get shorter this way. Okay, and it's going to get shorter this way. It's going to pull our head and turn it that way. So hopefully you can kind of envision why that would happen. And if you can't envision it, I encourage you to go ahead and put your hand on your sternocleidomastoid, allow it to contract, and see the movement that's occurring. Okay? So in addition to knowing and understanding the insertion and the origin, it's also important to understand that these attachments, they can either be direct attachments or they can be indirect attachments. And so in direct attachment, the epimysium is fused to the periosteum of the bone or the outer layer of cartilage. So a direct attachment is meaning to bone or to cartilage. Okay? Straight muscle to bone or straight muscle to cartilage. An indirect attachment, however, is when a muscle has a tendon or it has a aponeurosis, which is going to anchor muscle to the connective tissue covering of either a bone or cartilage or to the fascia of other muscles. So, for example, if the sternocleidomastoid had a tendon, then it would be considered an indirect attachment. Okay? All right. So we sort of started talking about skeletal muscle, and I have briefly mentioned these words before already once, just to sort of get you introduced to them and get you thinking this way. But let's remember that whole muscles, okay, so like your entire biceps muscle, for example, is full of fascicles. And fascicles, which we'll kind of move down to this next picture, fascicles within themselves have many muscle fibers. And a muscle fiber and a muscle cell are the exact same thing. Okay? And then what's important to understand that beyond just these muscle fibers, inside of the muscle fibers, we have, so I'm going to zoom in on this here, which is what you see in this bottom picture. inside of the muscle fibers, we have many myofibrils. Okay? So let's talk about that because I know that that's a pretty big progression here, and so I just want to make sure we're all on the same page. So this picture is representing a muscle fiber or an entire muscle cell, and you can see inside of it are many myofibrils, also mitochondria and sarcolemma and so on. Okay, so let's see what some of these things mean. All right, the sarcolemma is just the plasma membrane that's covering muscle fibers. Okay, super simple. The sarcoplasm is the muscle fiber cytoplasm. Okay, so that would be basically inside of this muscle cell. These myofibrils have a little bit of space in between them here and there. And that little bit of space in between them is where you would find things like mitochondria, which makes ATP, which is our major energy source. You would also find glycosomes there, which have the job of supplying glucose to mitochondria for ATP production. And then also you're going to find myoglobin within the sarcoplasm. And myoglobin is a pigment cell, so it's a red pigment, which is why muscle appears to have sort of a red color. And the purpose of myoglobin then is to store oxygen, because oxygen is actually also needed to make ATP. Okay, so as I've mentioned already before, and I'll mention again, each muscle fiber contains many myofibrils, and that's what you see being pulled out here. One myofibril in this top picture is being pulled out. Myofibrils are these very dense, compact, rod-like structures and make up about 80% of a muscle cell's volume. Okay, so about 80% of a muscle cell is myofibrils, and about 20% is all this other stuff that we were talking about. Okay? So in addition to these sarcolemma, plasm, myofibrils, I will next talk about the sarcoplasmic reticulum. And these are just really these smooth tubules that are surrounding each myofibril. Okay, so anything you see in blue here is considered sarcoplasmic reticulum. And sarcoplasmic reticulum's job is to regulate the amount of calcium that is entering the muscle cell. Okay, and we'll understand why that's important coming up in this lecture. Within, sort of embedded within the sarcoplasmic reticulum, we have these things called T-tubules. And the T-tubules are these lighter blue / whitish looking structures coming down through the sarcoplasmic reticulum. And they're formed by the sarcolemma up here diving deep into the cell. Okay, so you can imagine this sort of clear layer up here, the sarcolemma. If it decides to tuck sort of into the screen, we'll have these little indentations. All right, and these are our T-tubules. And their whole job is to increase surface area, which allows electrical signals to be reached by the deepest layers inside of a muscle fiber, so that all of the myofibrils can get this and receive the same electrical signal and respond accordingly. We'll talk more about this as we go on. Okay, so myofibrils. I know you guys are probably tired of hearing me say myofibrils already, but let's talk about them a little bit more. They're these densely packed rod-like elements inside of a muscle fiber. So this whole thing is a muscle fiber, which is packed of many myofibrils. And every muscle fiber contains thousands of these rod-like structures, which, as I mentioned on the last slide, is about where 80% of the muscle cell volume comes from. So something to note about myofibrils is that they are striated. They have sarcomeres. They also have myofilaments. And there's a molecular composition of the myofilaments. So we're going to talk about each of these things over the next few slides. All right, striations. Striations sounds like the word stripes, and that's because this literally means stripes that are formed by repeating series of dark bands and light bands, okay, along the length of each myofibril. So this is representing a myofibril. Okay, one myofibril inside of a muscle fiber. Remember, there are hundreds and thousands, perhaps even, of these inside of one muscle fiber. Okay, so myofibril is what you're looking at here. We have these alternating light bands and dark bands. The dark bands are the A bands. Okay, so you can sort of see this darker section. And so maybe I'll just draw, if I can, some of them in. Sometimes my drawing with my mouse is not that great, and so I'm sorry. I ordered a new computer, which has a touchscreen, so hopefully I'll get that soon. But in the meantime, just bear with my drawing. Okay, so we have the A bands, which are the darker bands. You can see here, this is a darker section. Here would be a darker section. Here's a darker section. And then we have within these A bands, right in the dead center, we have what's called the H zone. And this is a lighter region that runs, again, directly in the center of the A band. We also have what are called M lines. And M lines are the lines of protein that's going to run perpendicular right down the middle of the H zone. Okay, so for reference, let me just scribble this in. The H zone is this whole section that I just scribbled in yellow. The M lines are these three lines here, okay, that are running perpendicular to that H zone. All right. Finally, we have I bands, and the I bands are the lighter regions, which you can see in the screen here. And the I bands have directly in the center of them the Z discs, or also they are sometimes referred to as Z lines. And basically they are just sheets of proteins that are running down the middle of the I bands. Okay, and hopefully you can understand why they look like, or why they're called a Z disc, because they, let's see if I can do it, it's Z's all the way down. Okay. Kind of going along with that, if you're having trouble remembering the names of Z's and A's and I's and all of that, I just try to come up with random things. So I always say, you have a ham, and then everything else belongs to the other bands. So you can think of, let's see, dark ham. How about that? So we know the dark sections have H-A-M, dark ham. All right, and the H's, if I draw them on here, I'll draw them in yellow, why not? They're called H zones because they form an H all the way up and down them. Okay, you guys can see how the M line and the H zones, because the way the M line is running through here, it looks like H's, and why those look like Z's. All right, so just remember dark ham, and maybe you'll remember which zone goes with which. Okay. So earlier I mentioned in that histology slide of skeletal muscle that it was striated, okay, and I showed you the histology slide. And so what we see here are, if we just took a look at that and we zoomed in on it, you see how there's dark, light, dark, light, dark, and so on. That is representing those I bands and A bands. Okay? So hopefully that makes more sense now. Okay, so now we'll talk about the sarcomere. And the sarcomere runs from Z disc to Z disc. All right, so that is one sarcomere. These sarcomeres, these are the smallest contractile units or functional units of a muscle fiber. So what that means is that contractile, these are the sections that are getting shorter whenever a muscle contracts. Okay? Individual sarcomeres are going to align to end, basically kind of like they're lined up like a train. So here we would have another Z disc. Down here we would find another Z disc. All right, and so we would have another sarcomere, and we would have another sarcomere, and hopefully you can get the point that when these contract, the whole muscle is going to shorten. Okay? Sorry, my drawings are not great tonight. Doing it with my mouse is really difficult. Okay, so myofilaments is the next thing that I have to briefly touch on. And myofilaments include actin and myosin. Okay, actin and myosin. And they are present in this really orderly arrangement. And even though this slide only has words on it, I promise you, as we move throughout this lecture, you will become very confident with what actin and myosin look like. But actin are our thin filaments. They extend across the I band and partially in the A band, but they're anchored to the Z discs. Myosin are our thick filaments, and they run the entire length of the A band and connect to one another at the M line. Or I guess not really necessarily to one another, but they make their connections in muscle at the M line. The sarcomere cross-section is going to show sort of this hexagon arrangement of one thick filament and thin, 6 thin filaments. Okay? So, this is what I mean. Here we have our thin filaments. Here we have our thick filament. Okay, and here's the M line. And basically these bottom pictures here are saying this is what it would look like if you clipped through the eye band and we're looking down the length of the muscle fiber. Okay. All right, so let's expand upon this even more and talk about the molecular composition of these myofilaments. So as I have mentioned already, we have thick filaments, which are also called myosin, which, so if I took this thick filament, the one in sort of this peach color, and blew it up, it would look like this. And, excuse me, if I latched on to just one section, let's see if I can, I just took this section right here and zoomed in on it, it would look something like this that's down here, where there are two heads, a site for ATP to bind, the heads are next to a region that's really flexible, sort of like a hinge, and then we have the myosin tail. And all of this stuff is going to come up as we move throughout this lecture. Now, on the other side of the ring here, we have actin, which are our thin filaments. So remember, that's these blue helical looking lines here. And if we took that and we zoomed in on it, it would look like what is right here, what we have present. Now, essentially, if I took one of these, let's see if I can do a better job than that. If I took one of these little blue circles and sort of blew them up, that's what we're seeing down here. So those, this very bottom right section is showing you what actin subunits, basically what actin molecules would look like. All right, so that is down here. So that sort of takes care of actin then. So we're sort of done with actin. Now, sort of wrapped around with actin are these molecules called tropomyosin and troponin. And these molecules, so the tropomyosin is the more sort of, I guess, orange looking thin strand that's wrapped in this helical shape around actin. And then troponin, which is what you see in yellow, also have a role in sort of interacting with actin. And actin and myosin are going to interact, and we're going to learn all about that. In addition, we have some other sort of molecules that I'm going to bring onto your radar, things like titin, which is what you see up here in these little orange coils, which are essentially anchoring proteins. We also have nebulin, dystrophin, myomesin, and C proteins, and we're gonna briefly touch on some of those different things as we move along. All right, let's talk myosin just a little bit more. Myosin is what composes our thick filaments. It is made of 2 heavy and 4 light chains all wrapped together. The heavy chains are going to twist up and form this myosin tail, and the light chains are going to form the heads. Whenever muscle contracts, the heads actually are the portion that link the thick and the thin filaments together and therefore form cross bridges. Myosins are usually offset from each other, and this allows for this staggered array of heads at different points along the thick filament, which is kind of what you see happening here in this top section. They're sort of staggered all along the way, and this will make sense. I have a video coming up for you guys, and it will make sense why they look like that. So again, thin filaments are composed of the fibrous protein called actin, which are the blue subunits that you see here are the actin molecules. It's a polypeptide made of what's called globular or G-actin subunits. And these subunits have little active sites on them. And this is where the heads of the myosin is going to attach. G-actin subunits are linked together by a molecule that is going to form what's called long or fibrous actin, and then two of these actin strands, two of these F-actin strands are going to twist together and therefore form the thin filament. Okay, so we have a G-actin subunit. When we link the G's together, they become F. And when we twist them up into strands, they become thin filaments. So here's some of the other things, the other molecules that I had mentioned before. We have tropomyosin and troponin, which are those regulatory proteins that are bound to actin. And so here the sort of green looking chains are the tropomyosin, and then troponin are these little dots that are sort of connected to tropomyosin. They're sort of a, I don't know, burnt hazily orange color, I guess. In addition, we have an elastic filament, and that is what's going to make up titin, which I pointed out to you guys earlier, and I highlighted it with that color earlier. Titin basically holds the thick filaments in place. And so, additionally, because it's an elastic component, it helps recoil muscle after it's been stretched, and also helps the muscle from being stretched too far. In addition to these molecules, we also have a very important molecule called dystrophin. Okay, dystrophin. And dystrophin has the job of linking thin filaments to proteins that are in the sarcolemma themselves. Okay, so thin filaments, we want to link them up with proteins within that functional contraction unit. So Z disc to Z disc. And I'll talk a little bit in a second about why dystrophin is important. Finally, nebulin, myomesin, and C proteins, basically just bind filaments or bind sarcomeres together and help to maintain alignment within the sarcomere. Okay. So here's why dystrophin is important, because there are diseases that can present when dystrophin is a non-functional protein. So Duchenne muscular dystrophy, also called DMD, is caused by a defective dystrophin gene. So a gene is what makes a protein, and so if the gene is defective, then you can't make the protein properly. And so if dystrophin is not being made properly, or if it's not being made at all, then the thin filaments, the actin, can't be linked to the extracellular matrix, which is supposed to help stabilize the sarcolemma. And this causes the sarcolemma to tear more easily, which in turn allows more calcium to enter muscle cells, which in turn damages our contractile fibers. Okay? If you have damage, just like anywhere else, you're going to have inflammation follow afterwards. And eventually, because of this, the regenerative capacity, you can only fix something so many times. Eventually, it's just not going to be able to be fixed anymore. This is going to result in the muscle cells dying due to programmed cell death or apoptosis, and therefore leading to a decreased muscle mass. All right, so this is a disease, it's a very serious disease, and usually appears between 2 and 7 years old. Usually you can sense that the child is a 7-year-old that becomes really clumsy all of a sudden and falls sort of more than what you think is normal all of a sudden. This one does tend to be sex-linked, recessive disease, and so occurs almost exclusively in males. In addition to sort of the clumsiness and falling, some signs and symptoms would include proceeding from the extremities and upward. So it's going to start in the legs, and then it's going to move upwards along the body, and also can start in the arms and move basically from the appendicular part of the skeleton towards the axial part of the skeleton until eventually it reaches the head, the chest muscles, and the cardiac muscles. And once those muscles become affected, obviously survival is not possible anymore. Unfortunately, there's not a cure for this. There is clinical trials occurring. Every day we're hoping that we progress somewhere with those clinical trials and that someday there will be a cure. But at this point, even with supportive care, people with Duchenne muscular dystrophy can live into their 30s and beyond, but even in your 30s, it's still very, very young. So moving on from our molecular components, we're going to talk about the sarcoplasmic reticulum and also T-tubules. And so, as I had mentioned before, the sarcoplasmic reticulum is a network of smooth endoplasmic reticulum tubules that surrounds each myofibril, and most of them run longitudinally or along the length. So everything that you see again in blue would be considered sarcoplasmic reticulum. At the end, you'll have these what we call cisterns, which are basically going to form perpendicular cross channels across the A and the I bands, at the junction, I should say, of the A and the I band. And the sarcoplasmic reticulum, its whole job in life is to regulate how much calcium is inside of a muscle fiber. Okay? regulate the amount of calcium inside the muscle fiber. The sarcoplasmic reticulum also stores and releases calcium. This is going to be very important as we move forward in this discussion, and you'll understand why here in a little bit when I start talking a little bit more about calcium. Again, I had mentioned before, these T-tubules are these sort of white-looking lines that are formed due to the sarcolemma, that outer sheath, sort of diving deeper into the interior of a muscle. This, again, just increases the surface area. It also makes the lumen continuous with the extracellular space, and allows electrical nerve transmissions to reach deep into the inside of the muscle fibers so that all of the myofibrils can be stimulated at a sort of consistent and whole group rate, if you will. The tubules, their site of penetration is at the AI band junction between the terminal cisterns, okay? And so, again, the terminal cisterns are, not even sure. Let's see if I can get my pen out. Okay. Terminal cisterns would be just lateral to the T-tubules, and again, the T-tubules are these sort of white structures moving right down the middle, perpendicularly. Okay. T-tubules contain proteins that basically protrude into the space that's between the tubule and between the sarcolemma, and this helps act as a voltage sensor that is going to change shape whenever electricity passes through. Okay, so whenever nerves are transmitting signals in the form of electricity, which is what they do, an electrical current is what they're giving, these tubules are sensing that. And the sarcoplasmic reticulum, these cistern membranes, these sort of blue structures that you see, they also have proteins that are going to protrude into the intermembrane space, or the space between the tubule and the muscle fiber sarcolemma. And basically, these control the opening of calcium channels. So when an electrical impulse passes by, these T-tubule proteins change their shape. Okay, so the proteins that are associated with these white bands, they're going to change their shape. That's going to cause the proteins on these sarcoplasmic reticulum membranes to also change shape, and that's going to cause calcium to be released into the cytoplasm. Okay, so just to kind of give you, I guess, more of a picture of this. Okay, if I were to encircle this whole thing, right, because this, remember, is one whole muscle fiber, right, and we have many of these myofibrils inside. Essentially, what's happening is, let me just fill this in. Alright, so if this is the outside of the muscle, it's going to be really sloppy. I'm really sorry. It's kind of a mess. Alright. And then anywhere in yellow is going to be considered cytoplasm inside of this muscle fiber. Because remember, the cytoplasm is where we're going to find things like the mitochondria. So basically what's happening is that whenever there's an electrical signal passing through, whenever it comes through the T-tubule, T-tubule changes its shape. Okay, so it's coming through, electrical signal is passing by. Here we go, it's passing by. The T-tubule is changing shape. That's changing shape. The sarcoplasmic reticulum, the blue, is changing shape, and calcium is going into the yellow space. Okay, calcium is going into the yellow space, into the cytoplasm. Okay, so hopefully that makes more sense. I know my picture is not great. I'm really sorry about that, but hopefully you get the picture, literally. This is just a summary chart of all of the different things that I've told you in terms of structurally and organizationally mapping out skeletal muscle. Okay, so before I go on to the sliding filament theory, I am actually going to start a new video. So everything we've covered in this video was more related to anatomy. And now starting at learning goal 4 through learning goal 9, it's going to be about muscle cell physiology. And then the remaining learning objectives starting at learning objective number 10 is going to be whole muscle physiology. So we're going to do this lecture in 3 parts. That way you don't have to sit down and watch it all at one time if you don't want to, and you can re-spark your interest. So I'm going to stop this here, and in my next video, I will pick up with muscle cell physiology.